Good evening, everyone. I'll just make my presentation very short. So
Yeah.
I'm Belen Lumeran from University of Technology, Bahrain. I'll be sharing my results for phytoplankton as biosensor for climate change. I'll be doing a short introduction, a statement of problem, literature review, methodology, results, conclusion, and recommendations. So this is more about what is really phytoplankton. So in the photic zone of the sea are phytoplankton, which are microscopic organisms following the dynamics of currents. The vital function is the natural removal of carbon dioxide from the atmosphere. Their presence exert local effect associated with climate change. Why phytoplankton can be used as biological sensor for climate change? Because they rapidly respond to changes in environmental factors. So I'll be measuring, actually I measured the water temperature, the surface water temperature, pH and salinity. Why only the surface water temperature? Because they are said to be found on the photic zone or surface dwellers. Climate change as a natural phenomenon may exert impact both the macro and micro components of the marine fauna, specifically the plankton biota. Hence, this study was conducted using phytoplankton as biosensor for climate change. The general objective of my study is to use phytoplankton as biosensor for climate change, specifically to conduct microscopic determination of species composition, calculate total abundance, find out significant differences in the calculated abundance, describe the total population as influenced by environmental factors, and statistically analyze to assess the role of phytoplankton as biosensor for climate change in a local scale. Local scale is only, of course, in the Bahrain scenario, because they're actually asking, are we experiencing climate change because of the drastic change in the temperature? So just a synopsis on the literature review, just the recent literature. Planktons are generally found on the vast sea, and they affect the structure and composition. Climate change related conditions in the ocean water will change phytoplankton population. There are climate-induced factors that stimulate phytoplankton growth. Organisms which are sensitive to climate change show dramatic changes in distribution, like the phytoplankton community. Marine planktons cannot track optimal temperature, hence assemblage undergoes restructuring. Changes in environmental factors may be short-term or long-term that changes the species composition. So I use experimental research based on the microscopy data for 5 years, commencing from 2015 to 2019, using the overall population based on total abundance, as may be affected by physicochemical factors, limiting only to surface water temperature, salinity, and pH as well. So I conducted the monthly sampling from 2015 to 2019. In just a small part of the Bahrain Sea using motorized boats and the following horizontal tau. I used actually the ecological field kit for in situ determination of water temperature, pH, and salinity. Then after that, I did quantitative microscopic analysis in our laboratory, and statistical analysis using ANOVA, or analysis of variance, to determine the differences in the total abundance relating to population count of the phytoplankton. And later, I used post-Hoc Turkey beta to evaluate any significant differences in the various pair of means as determinant of the total abundance. And the response of phytoplankton was analyzed using Pearson coefficient of correlation. So in my short study for 5 years, only 18 phytoplankton species were identified. The total annual abundance is increasing annually, with a total population for 5 years of only 8,864. So I only covered about 15 kilometers from offshore. So ANOVA revealed significant difference at 0.05, which means that phytoplankton respond to overall environmental changes, although this response is still a natural biorhythmic response. Post-Hoc Turkey, HSD, on the other hand, revealed insignificant difference such that same overall response to the measured environmental factors such as the surface water temperature, pH, and salinity. In situ physicochemical monitoring showed average surface water temperature of 35.5 degrees Celsius, pH of 8.0, and salinity of 40.35 parts per thousand. So these values, these averages are within the tolerable limits. So result of Pearson coefficient of correlation showed strong positive correlation. So I will not be discussing one by one. So what does the result mean? So it means that the phytoplankton community showed a positive response. However, there is no differences in terms of the total abundance annually. So it means that there is no change in terms of the population annually, such that there is no significant difference in between the means of the annual population. So based on this, the following are my conclusions. The phytoplankton assemblage is abundant as indicated by the total annual microscopic count. The measured environmental indices relating to surface water temperature, pH, and salinity exerted direct or indirect impact on the population of phytoplankton. The response of the phytoplankton assemblage to agents of climate change indicates biological sensing capability, thus can be used as biological sensors. Statistically, the varying phytoplankton abundance is a biorhythmic response to microscale climate change. A long-term monitoring is one of my recommendations of the microbiota of photic zone be conducted to fully assess the abundance of phytoplankton. Quantitative analysis of the sea water focuses on grazing productivity be conducted to validate the direct and indirect effect of the identified phytochemical attributes. A thorough study on the biological sensing capability of the phytoplankton be further experimented in a laboratory setting. Further study on the biorhythmic responses to microscale climate change be conducted covering a wider area of interest to better assess the abundance of phytoplankton and other species that may be screened and microscopically identified. An in-depth study be undertaken on the anatomical and structural attributes of the phytoplankton to fully determine the biological sensor capability based on the organism's responses to the indicators of climate change. Thank you very much for sharing your time. Should you have any further questions, because this is just a synopsis of my result, you are free to. Thank you very much.